Welcome, my dear students. Today we step into one of the most fundamental concepts of biochemistry, the peptide bond. How do tiny amino acids join to form long chains that become functional proteins? And how can a single bond influence a protein structure and its stability? In this class, we will uncover how the peptide bond forms, why it is unique and how its special characteristics support the entire architecture of proteins. So let's dive into the simple yet powerful chemistry behind this essential biological bond. So first of all, we have to know what is a peptide bond. It is a special type of covalent bond that is substituted amide linkage that joins two amino acids. It forms between the carboxyl group of one amino acid and the amino group of the next amino acid. So how this peptide bond is formed? This involves dehydration or removal of water between the alpha carboxyl group of one amino acid and the alpha amino group of another by removal of water, that is which causes dehydration or condensation reaction. It is an example of condensation reaction. So common biological processes occur during protein synthesis. Oligopeptide, it contains a few amino acids joined by peptide bonds. Polypeptide contains many amino acids linked together. Protein, polypeptides with molecular weight greater than 10,000, and all proteins have one N-terminal residue, that is the end with a free alpha amino group, and a C-terminal residue, that is the end with a free carboxyl group. This is the image showing the formation of peptide bond between two successive amino acids. End of a polypeptide chain, N-terminal or amino terminal, which has a free amino group, and the C-terminal or the carboxyl terminal has the free carboxyl group. So the structure of the peptide bond, which was proposed by Pauling and Corey's findings: partial double bond character. Peptide bond CN bond is shorter than a normal CN bond. So due to resonance between C double bond O and N bond H, it results in oxygen becomes partially negative charge and nitrogen becomes positive, partial positive charge. So a dipole is formed in between the structure. So planarity, 6 atoms including the CN bond, that is C alpha C O N H C alpha, lie in one plane. That is C double bond O and bond NH bonds are trans. Uh, Atoms in the peptide bond are coplanar, that is they live in the same plane and it form a rigid plane. Restricted rotation around the peptide bond. CN bond, that is a partial double bond, or the peptide bond cannot rotate freely. Rotation allowed only around N C alpha bond or phi bond and C alpha C bond or psi bond. This is the image of Pauling and Corey. He firstly described about the peptide bond structure. This is the formation of the peptide bond in between two amino acids. One water molecule is released. This is a dehydration reaction or condensation reaction. Now you should know the peptide bond characteristics. Peptide CN bonds cannot rotate freely because of partial double bond character. The rotation occurs only at NC alpha and C alpha C bonds. Backbone consists of rigid planes with rotation possible at C alpha, and consecutive planes share a common rotation point at C alpha. So the bond angles: phi angle, that is the rotation angle around NC alpha bond, and psi angle, that is the rotation angle around C alpha C bond. Both phi and psi are 180 degree in the fully extended polypeptide conformation. Ramachandran plot: phi and psi angles can range from minus 180 degree to plus 180 degree, but many values are restricted due to steric hindrance in the polypeptide backbone and side chains. Graphically, the Ramachandran plot graphically represents allowed phi and psi values for the polypeptide chain, and this plot was introduced by G.M. Ramachandran to study protein conformations. And with that, my dear students, we conclude our journey into the world of peptide bonds, the tiny yet powerful links that form the backbone of every protein. From how this bond forms to why it is a rigid and planar, we now understand its central role in the protein structure and stability. Whether you are preparing for UG or PG or NET life science, mastering the peptide bond builds the foundation for understanding protein folding, enzyme action, and structural biology. Keep exploring, keep learning, and let your curiosity guide you deeper into the world of biochemistry.